as we know, the current technology for high-throughput screening profiling in the market is there are, we can do high-throughput screening at the DNA level. We can do high-throughput screening at the on the protein level, but we cannot, or not say cannot, at this time we don't have a good one that can do high-throughput screening profiling for the sub-cellular localization profile at this level. Basically, say we can determine the localization of thousands of protein from one experiment. Basically, that's what we're shooting for. And the technology we have developed is called DAMA staining technology. It's an abbreviation of Dissociable Antibody MicroArray. So it's a kind of it's a high-throughput platform for the profiling sub-cellular localization and expression profile, and it's basically a combination of immunostaining and the protein microarray. So, and it's a unique technology for that. So this is the summary of the technology. So basically, we'll grow the cell and fix and permeabilize the cell on the cover slip, just like we do the immunostaining. Actually, after this, we add the primary antibody and then add the secondary antibody for the immunostaining. That's usually measured. And at this time, we add 400 to thousands of antibody to the target sample from a membrane. And now basically deliver the antibody to the specific location without diffusion. And then if we can add the secondary antibody, either fluorescence conjugated secondary antibody or HRP conjugated secondary antibody, then we can determine the protein expression or subcellular localization profiling. So we can get some spots like this. And so the difference between this antibody array and the captured antibody array on the market is that the sample is different. So the sample for the captured array is a slice set, and here the sample is the fixed cell, is the whole cell. So we can get both the information of expression level and the subcellular localization level. So basically that's one. And then this is the procedure. So we do the sample preparation and the DAMA staining, basically make the antibody array preparation, and then do the experiment for the DAMA staining, and then we get the imager, and then data extraction and data analysis. So first I will tell you, so to develop this technology, we work with two steps. First step, we determine the protein expression profiling by this technology. So at this time, this is the design of antibody array. At this time, it's large. It's kind of a 3 centimeter by about 4 centimeters. So this is the antibody array. We put about 400 antibody on the membrane. And this is the apparatus for the experiment. So at this time, this is the first time we do it. So we grow cell on 10 centimeter culture dishes. These are cell lines. We grow them and fix them and permeabilize them, and then add the antibody array. Oh. And this is antibody array. So this is the cell will grow on the cell culture dishes and fix and permeabilize. Oh, I should bring my. I may have. So cell will grow fixed and permeabilized, and this is antibody membrane that we put on top of the cell and incubate, basically say, add some pressure on it and incubate for 2 hours. And after that, we remove the membrane. So this is basically cell culture. Cell will grow on the cover slip, this membrane fixed for 2 hours, then remove the membrane. For the reason we don't know, the antibody was delivered to the membrane without diffusion. So if we want to say it, then maybe it's an antibody-antigen interaction is stronger than antibody-member interaction. So maybe that could be the reason, but we don't know the reason. So basically, then at this time, if we add to alkaline phosphatase conjugate the secondary antibody, then we can determine the detector bonding antibody. So basically, this is the result comparison of protein expression profile between two breast cancer cell lines, this one and this two, and the intensity of the spots corresponding to the amount of the specific protein. So by this method, the first application is we try to identify differentially expressed protein in breast cell and breast cancer cell lines. So this is standard. We use 10 cell lines, 3 normal, 7 cancer, and these are cell lines purchased from ATCC. And do the DAMA staining, do data extraction and analysis and prediction, and then do Western blot to validate it. So these are the experiment data of the 10 breast cell lines. The first 3 are the result from 3 normal cell lines. The last 7 are the result from 7 cancer cell lines. And then we wrote a program to really do the data extraction and data analysis. Among the 360, around 400 proteins, we identified 10 proteins whose expression level are differentially different between cancer cell lines and the normal cell lines. And then we do the Western blot to validate them. For those proteins whose expression level will predict lower in cancer cells, we cannot confirm them. Maybe we think because we only have 400 proteins, and most of them are cancer-related proteins, so we don't have a good statistical system. So, and among the 7 cell lines whose expression level are highly expressed, we confirm in cancer cells, then in normal cells, we confirm 5 of them. And then this is the result. So each line corresponding to 1 antibody, and the first 3 lanes corresponding to the normal cell, the last 7 corresponding to the expression level in the cancer cells, and the last lane is acting as a control. And we also further test among those 5 proteins, we further examine the expression level by the tissue microarray. And our preliminary data show that we confirm like 3 of them. We don't confirm them. The 3 of them show the difference between cancer, between tumor tissue and benign tissue. So next one is try to identify, use this method to do the sub-cellular localization profiling. And it's a similar apparatus, but at this time, the sample is the cover slip. Where the tissue, the cell line sample will grow on 2 times 2 centimeter cover slip. So after grow them, fix them, permeabilize them, and the membrane, the antibody membrane was put on top of it and incubated for 2 hours, then remove the membrane. So at this time, we redesigned the antibody array. At this time, it's a smaller array. It's a 1 centimeter by 1 centimeter array, and the first time it contains 96 antibodies. And on 4 corners, we put a red spots there. Those are antibody against the histone, so that we can identify the location under the microscope, so we can tell the orientation of the cover slip. So similar procedure. The only differences here is we add the fluorescence to conjugate the secondary antibody. So at this time, if we can move the microscope to these spots to take 5 pictures, then we can get the image of this 5 protein. And move to these spots, get 5 pictures. So basically say, we can move the microscope to the specific spots, then we can get the image of that specific protein. This way, we can get the image of hundreds of proteins from 1 experiment. And we use a standard microscope. This is one of the microscopes at Louisiana State University. And the only difference is that this is a motorized-driven stage. The stage was motorized-driven, controlled by computer. So we need, and then the slider program, the program to control the microscope wrote a spatial module for us. So anybody can do the experiment as long as they have the microscope and maybe purchase additional module. So this is the antibody design. On the four corner, we have a histone antibody. So that means under the microscope, we tell the computer where is up left corner, up right corner, and tell the computer of the four corners. So the computer can define the orientation of the cover slip. Then the computer will move to the A1 to take 5 pictures, move to A2 to take 5 pictures, and so on, so they can get the pictures of all 100 proteins. So these are the some examples of the images. So this is the image we got at the low resolution. You can see it's really, the image looks like a spot and without a diffusion. And this is a high resolution. So the images are obtained by this method are similar to those images obtained by individual immunostaining. Then we wrote a program for the data analysis. So this basically say we're talking about, if we talk about 400 proteins, each protein have 5 images. So we talk about 2,000 images per samples. If we have 10 samples, so we're talking about 20,000 images. So we wrote a program to do the data analysis. Basically say this is input data, input 20,000 images into the database, and write a program to adjust, maintenance those images, and adjust the quality of images. So there are 2 ways to view them. This way is compare the image of a specific protein among different samples. So this is one. And this is another one, is the image of different protein in one sample. So each spot corresponding to the image of the specific protein in the sample. So we also use this procedure to identify unique sub-cellular localization marker in the breast cancer cell lines. So same thing, and we have, at this time, only 2 normal cells can be grown. So we use 2 normal cells, 7 cancer cells, and do the DAMA staining and compare the sub-cellular localization pattern of 360 proteins between normal and the cancer cells. Then identify protein that with unique sub-cellular localization between cancer cells and the normal cells. So this experiment, at this time, we use 4 different arrays. Each array contains about 96 antibodies. So because this is the first time to do it. So we use 4 arrays. And this is the sub-cellular localization profiling of 360 protein from one sample. This is a benign sample. And each imaging corresponding to a specific protein in that sample. So basically, this is the published data. Looks like beautiful. Then that information is too much. And then what we try to, for each imaging, we define a sub-cellular localization code. So basically say, we don't want to show the image. We just say, well, does this protein localize? At this time, we use 3 code. Nuclei, for example, this is green. We have a DABI there. If the image is co-localized with the overlap with the blue, we say this is a nuclear localization. If it's exclusive from a nuclei, we say this is cytosol localization. And if the localized in both places, we say it's both places. So basically, for each imaging, we assign a sub-cellular localization code, and then this is published. This is the summary of the sub-cellular localization profiling of all of the experiment data. So for each vertical column corresponding to a specific protein. So for each horizontal line rows corresponding to a specific sample. So there are 2N, it's 2 normal cells and 7 cancer cells. So the color code for the sub-cellular localization is red is a nuclei, green is cytosol, and yellow is both, and black is unclear. So basically, that's data. And from those data, we identify one protein, cyclin B1, whose expression level are different, whose subcellular localization are different between cancer cells and the normal cells. So those are the, in cancer cells, they're in the everywhere, in normal cells, they're in everywhere, but in cancer cells, they're in cytosol. So this is, and also we identify that their expression level are different between cancer cells and the normal cells. And from the image analysis, those are validated by Western blot. So this is, and next one, what we're doing is to move the antibody array from 100 antibody per membrane to 400 antibody per membrane. So this is data. So we put a 400 in a membrane in a 2 centimeter by 4 centimeter slides. We grow the cell on the 2 centimeter by 4 centimeter slides. We use 2 normal cells and 3 cancer cells. At this time, we just do the test. We don't have enough samples. So basically, this is summary result of the image of 400 proteins in one of the prostate cancer cells. So this is a summary of subcellular localization code of 400 protein in 5 prostate cells. So at this time, we have different color code. So some are membrane, some are nuclear, some are cytosol, some are spots. So basically, because the image is a little bit, we can tell more, we can see different pattern now. So from this analysis, we identify one protein, GLK1. And this protein has it's localized in membrane in some of the cancer cells. So these 3 are normal cells, these 3 are cancer cells. So this membrane, this protein localized in 2 of the membrane of 2 of the cancer cells, but the localized in cytosol of normal cells and other cancer cells. So we don't want to make a conclusion at this time, but at least we validate those observations by the individual immunostaining. So this is the result we have. So future direction, it's this is the preliminary data on tissue array. So this method can be used for the tissue samples, and this is the result comparison between the image of 3 protein between 2 different samples. These are the imaging obtained by the antibody array and by the DAMA staining method. These are the images obtained by individual staining. Basically, say we can get the same imaging as determined by the individual staining. Another thing what we're moving forward is to determine the expression level by fluorescence. So, and use the internal control. So we have 2 references. One is the red, and one is the green. So red basically is used as a reference. For example, use actin as a reference. And these are the 2 different ones, and this is one experiment that we're doing quality control. So basically, we can have a good statistic. Basically, if we repeat it, we can overlap. We can have a good reproducibility. Another one is similar to the in-cell ELISA array in the market. So at this time, we're not just doing one ELISA. We can do maybe more than 10 ELISA from one experiment. So for example, at this time, we think about to target 24 wells sample. Basically, say the cell will grow on 24 wells and treat it with different either drug or whatever they related to the customer, and fix the cell and permeabilize cell and ready for immunostaining, and add our array to it, and add a secondary antibody to detect the bound antibody. Then we can detect the expression level of interest in the protein. So at this time, we think about maybe 10. So another one is extended this chip from 400 to 1,000. So we can do this. This is the design to make the array, and from 2 centimeter to we can make the put a 1,000 antibody on the membrane of 2 centimeter by 4 centimeters. So for the interested, for the customer, they can grow the cells on 2 centimeter by 4 centimeter slice. And then they can use this array, then we can get the image of 1,000 protein. Then another thing is to try to identify the sub-cellular localization of the interested protein. So for example, one color is the stained with the protein of custom interested protein. On the red, then we have use our antibody array. So different antibody, different spots corresponding to the mark of different organelle. For example, for the nuclear marker, plasma membrane marker, cytosol marker, early adenosine marker, mitochondrial marker, and so on. So then if we can put this together, we compare the red and the green to see where does the green co-localize with the marker. So we can tell where, the interest protein, how much % of interest protein is in different organelle biomarkers. So this can be used for the trafficking study. So that's the thing. And finally, this is acknowledgement. These are the group people in my lab when I was at LLC, and this work was done mainly at Louisiana State University, and right now I'm at ATCL now. And this was received grant support from NIH, Susan G. Komen Foundation, and right now we're collaborating with FDA, Pfizer, and Biotronics, and this is the company we're kind of working on, Proteomics. Yeah. So, and thank you.
Now the talk is for open discussion.
Yeah.
You're talking about sub-cellular localization in cell lines. You found many differentially localized proteins in cancer versus normal cells, right?
Yes.
And have you gone to actual tissue sections and then immunochemistry to see the same thing in actual cancer tissue?
No, we haven't on that stage yet. So right now we're more like develop this technology and as a platform so that the people, the biomedical research field can use it. So right now we're more like a platform developer stage. Yeah. Yeah.
I really appreciate your talk. I think this is something that is underappreciated, the importance of cellular localization.
Yeah.
Have you looked at, for instance, in your prostate cancer models, we know that androgen receptor cycles from the cytoplasm to the nucleus. And it's nuclear AR which drives these expression of genes that cause low stereotypes in patients who have prostate cancer. So have you done controls where you have cells that are dosed or undosed with DHD, the hormone, and actually show changes in localization of androgen receptors? So the same thing with estrogen receptor, those kinds of.
No, we haven't. So next one we try to do is basically use those localization marker and then find the right project to really to do it. Yeah, that's what we're trying to basically find a specific project and then dynamically treat the cell, fix the cell after treatment at different time, and then quantitatively study. Basically say how much % is in this organelle, in this. Yeah, so that's what we're trying to do. Yeah. So that's what we're, yeah. Thank you. Okay, good. Okay, thank you.
Thank you, John. Please.
Oh, thank you.